I want to thank everybody for joining us for today's live webinar entitled Learn How Discovery Data Technology Enables Faster Discovery Research. Today's presentation is brought to you by Certara, and it's also sponsored by Asterix Technology. Asterix is running a series of webinars now. We've been doing this for actually over 12 months, where we're bringing together thought leaders in the lab informatics and laboratory technology space to share their vision on where laboratory technology is going, and this is part of that series. We can go to the next slide, please. Before we get into the presentation, if you're not familiar with Asterix, if this is your first time attending one of our sessions, let me take a moment and give you a quick overview of who we are. We are a professional services organization in the lab informatics and lab technology space. We successfully deliver highly specialized services, process technology, and outsource services to fundamentally transform how science-based businesses operate. This is all in an effort to improve scientific outcomes and the lives of people everywhere. Some quick stats about the company. We were established in 1995 and are privately held. We originated as the IT division of APBI Life Science Research Organization, and we operate from 7 offices in the United States and Costa Rica, with our headquarters in Red Bank, New Jersey. The type of customers we help are typically going to be everybody from Fortune 1000 life science enterprise, chemical companies, CPG companies, government research institutions, or mid-sized enterprises, basically organizations with large and fast-growing IT or outsource or compliance needs. We go to the next slide, please. A quick snapshot of the full suite of services we provide, and what we like to tell people is we provide services that span the entire life cycle of scientific data systems. This is everything from helping companies understand the type of technology they need in the first place. That's through business process analysis and the architecture and design of what your tech stack's going to look like. From there, we can go through specifically helping you choose and implement specific laboratory technologies or informatics technologies. And on the back end of it, then we can help with everything from validation to providing ongoing services for these systems to even providing staffing for these systems or staffing in general that you may need in your laboratory. So that's a little bit about Asterix. I want to now take a moment and introduce our speakers for today and turn it over to him. David Lois is going to be joining us. He's the Executive Director of Scientific Intelligence at Certara. I'll let David tell you a little bit about himself, but he has a BA in chemistry from Oxford, as well as computational chemistry from Oxford, and he has a postdoc in Washington University in St. Louis. So David, you've got a lot of really cool background stuff in here. I'm going to let you tell the folks a little bit about yourself and then let you pick it up from here.
Sure, thanks a lot. And firstly, thanks for inviting us to speak. This is a subject close to my heart, as you can see from my brief bio. I have a history in computational chemistry, but for the last 10 years, I've been working in the informatics area. Certainly realized that if you don't know where your data is coming from and you don't know how it's got aggregated, then you've got to be very careful about how to use it. So having said that, my responsibilities at Certara for the scientific informatics areas, and at the heart of that is a product called D360, which we've had out on the market for about 10 years. We deal with all kinds of organizations, from the very large multinational pharmaceutical companies down to small biotechs. So I just wanted to talk to you a little about what we see as best practices and data utilization, talk a little about how D360 is applied to that, and give you a brief demo of the system. So just to begin with, so what are we talking about here? So D360 is really a piece of software designed to help scientists get to their data. And that includes getting to their data when that data might come from multiple sources. And the key aspect that we want to enable here is that this is self-service access on the part of the scientist. So if you want to go and see your project data, we want that to be available essentially as a one-click operation. But at some point, there has to be a building of that data view that you want to see. And with D360, scientists can do that for themselves. And that might be a very simple view, as you see here, with just a color-coded spreadsheet of, in this case, chemical compounds. But it might be something a little more advanced. So we see some data visualizations, but also structure and sequence analytics. So we're not just delivering the data, we want to help scientists get insight into what that data actually means in order to make prioritization decisions, and also to design new compounds, new sequences, and the like. So we're very much focused on delivering that scientific data. More recently, we're also enabled not only the delivery of data to scientists, but delivery of data to other processes as well. So now we can deliver that data. We can provide analytics on top of it. None of this discovery stuff is done in isolation by individuals. It's done in teams. And so all of these capabilities can be shared amongst teams. Data can be added in the form of annotations in order to share insights, thoughts, experimental progress, and the like. So D360 is really here to support research workflows. Just to be clear here, D360 is not a registration system. We don't capture data within the system. We don't register compounds. We don't capture assay data. We are not an ELN. But D360 plugs into all of those types of systems. So we can search data across one or multiple data sources. We can provide data views from many different perspectives to address the workflows of different scientists, whether that be chemists, biologists, managers, and the like. And we configure D360 to look across those data sources so that users can build their own queries, build their own data views, and then share those out with other team members. What are our goals for D360? Well, first and foremost, to serve the needs of the scientists. It's been really interesting over the last decade, seeing and understanding how people get to their data, what they want to use their data for, and the difficulties involved. So what we need to do here is first and foremost, satisfy those needs. And we can do that by providing a very extensible system. So D360 is very highly configurable and customizable as well, but configuration is really the key. So we configure D360 to look at all the sources of data that a user would want to see and bring together. And then we can configure those data sources in a way that changes the perspective on data from how it's stored in a physical data architecture to how a user wants to see it, where they think about a scientifically logical data architecture, where you're not thinking about tables and databases or web services, but you're thinking about compounds and assay results or sequences or progress through experiments. As I mentioned before, we started this program really looking at the idea of the medicinal chemist, and when we did that, we saw that there were many different kinds of data that people wanted to see, whether that was scientific or logistical. But once we've configured the data, we can now present it from many different angles. So if you're a biologist running an assay and want to understand the QA of that assay, that's quite a different data view, where you're looking at a lot of data on maybe a few control compounds rather than many compounds and many assays. And D360 allows all of those pieces. And a really key aspect of this is keeping the costs of maintenance of this whole system down. So D360 itself requires essentially very little to no maintenance on a running system. D360 will automatically make new data available, and that includes looking for when new assays and new tests come online. So the actual operation of the system is essentially minimal. You don't need specialized IT to configure the system all the time. You don't need specialized users to build queries or dashboards. The user community for D360 is essentially self-service. Just to show you the breadth of where we've got. So we did start in the small molecule discovery area, and the kind of things we think about there are chemical compounds, assay results, what are the batches of those compounds, what is the logistics of the situation to how much of a compound might be left. Because as you go through a discovery process, it's not just the science that matters, it's the logistics also, because you need to make progress at a certain speed. And even though you might want to prioritize compound A over B, if you have more compound B available, that might be a more sensible decision to drive that forward. But since we started in small molecule discovery, D360 at its heart is actually quite a general informatics application. And so we can add additional tools for the representation of different kinds of entities. So over the last 2 years, we spent a lot of time on sequence descriptions, and that's extended our footprint into peptide discovery, antibodies, but also many other types of new modality, oligos, ADCs, and all of that information can be brought together. So whether your project might be on antibodies or whether it might be on small molecules, you can use the same application, the same data visualizations, the same treatment of assay data, but with specialized tools available to treat sequences versus chemical structures. And we've gone actually even further than that, and we've dealt with preclinical animal safety data as well. So that means looking at preclinical safety studies. And here in this area, your rows in a spreadsheet, for example, may be an animal, maybe a study group, as opposed to a chemical compound. As mentioned earlier, we also want to try and address different workflows for different scientists. So if you're designing new substances, for example, a medicinal chemist, the standard view would be a compound-centric view. But as you get into later stages and you start in vivo testing, then it becomes more important to have a batch-centric view, because impurities or other items within those batches will affect the outcomes of those experiments. If you're a biologist running an assay or executing an assay, you might want quite a different view where you're understanding how control compounds act over time. And so once we've configured the data, you can chop and change the data view that you want between these different entities of interest, essentially just by building a new data view. But the capabilities are all present. In terms of scalability as well, so we started off actually with at the high end. So D360 was initially developed in combination with Wyeth and with Pfizer. So we have some customers who have many thousands of users. But we scale right down to the smaller end as well. So we have our smallest customers have about 10 users. So very widely deployed, including 5 of the top 10 pharmaceutical companies. We are deployable in a variety of situations. So historically, if you go back to the beginning of this, everything was on premise. What we see now is there's a substantial amount of people moving towards private cloud. So we can deploy there. And we also provide a software as a service option where data sources are appropriately accessible. Also, over the last 10 years, we've developed standard configurations against many systems. So we have arrangements and partnerships, for example, with Siligence or CDD Vault to be able to drop D360 on top of those data sources, essentially in a matter of a few hours. And we also have standard connectors into many other standard commercial systems. But on top of this, we also can configure and most frequently do with our larger customers D360 to access data sources which are essentially bespoke, built for that customer, so data warehouses and data marts. So very versatile into how we can deploy, very versatile into what kind of modalities and what kind of scientific workflows we can support. So let's just focus a little on this. So we kind of said what D360 can do, but why is this important? So if we look for a second just at a standard small molecule research cycle, essentially this is made up of design, make, test, and analyze. And data flows in different ways into this. So when you're making a new substance, you're going to synthesize that substance or purchase that substance, but you'll register it. And so we've got a data in procedure. And so now you've got an ID, an associated molecule, a new batch, and a load of data about that molecule. When we test, then we go to assay execution. And here we're just showing a couple of screenshots out of CDD Vault. And so now we're going to get IC50 values, particular assays, maybe measured under different conditions. One thing that you'll generally find in common with all these data capture tools is they're really good at actually capturing the data. They're very good at presenting the data back in the way it's being captured, but not so good in presenting it and helping analyze the data in order to make research decisions. And that's where D360 comes in. So regardless of whether your registration database is one system and a different one from your assays, or they're all the same, then we can bring all of that data together in D360. Essentially, we can save that as a project data view. Users can then, as a single click, access that data and bring to bear a number of analytical tools. It might be basic data visualization, but it might be more advanced capabilities. And that can be used for prioritization, but also to feed into the design stage. And so D360 allows you to sketch new molecules, to enumerate different substances based on what you find from your data. So key capabilities here really are self-service access for the scientist. We want to combine the tools into one application. So not only do you get your data, but you retain the context of that data for analysis. And all of this without the need for an army of IT people underneath. So this looks at one design, make, test, and analyze cycle, but that's not how this research works. It's not just one cycle. If we're trying to improve the bioprofile of a substance, whether that be a small molecule, an antibody, an oligo, etc., there are actually a whole array of these design, make, test, analyze cycles that go into producing the substance that is ready to go into the next stage, which is going to be preclinical and animal testing. So this is what we want to do. So what we're trying to attempt to do with D360, and what we've been very successful in doing, over 5,000 users worldwide globally, but we want to make this whole process more efficient and more data driven. And that means we want to really provide real-time data access. So as soon as data is available within a source system, as soon as it's been captured, that needs to be available to the scientists so they can make that decision there and then, and not either have to wait or make a decision that does not include that data when it really is actually available. We also want to deal with very consistent treatment of data as well. So when we're looking at assay results, the compound may have been measured many, many times, and we want to present that data in such a way that we understand what are those measurements telling us? What is the mean or geometric mean of that IC50 value? How do you calculate such things when some of the data in the assay might be greater than or less than or even textual values? And then how do we make that consistent over different modalities or over different compounds? So those are the key capabilities. And really where we want to get to is say, look, each of these design, make, test, analyze cycles is shorter because it's much easier to assemble and get to all the data. There's no manual data manipulation. There's no moving data between applications unless you really want to go into something very advanced. So we want to shorten each cycle, but we also want to reduce the number of cycles by making them consistent. And so the goal here is to speed up the whole discovery process by making better use of that data. So one of the workflows that we really look at a very high level perspective, the key workflows really are thinking about prioritization. So is substance A better than substance B? Okay, which should we move forward with in terms of chemotype or sequence type? It's also looking at substance design, understanding what it is about these substances that makes some of them have positive, good bioprofiles, some of them have less good bioprofiles, finding out which groups of substance are best to optimize and which properties need to be optimized. But also getting into more logistical, quality and scientific workflows beyond this. And so that could just be ad hoc data analysis. I look at my data, I get an idea. Now let me search the wealth of data that we may have available in order to prove or disprove that hypothesis in order to drive that project forward. So this is where we're aiming. But I think it's very important that we've been successful in this because of the way that we develop the product. And we develop the product in very close collaboration with our customers and users. At a strategic level, we have a customer steering committee, and that customer steering committee kind of gives us a strategic guidance on the elements that are most important to them. And the steering committee is made up of 10 of a representation of our customer base and rotates on a regular basis. But that group meets twice a year. The current priorities provided to us are the biologics analysis tools, so thinking more about understanding how sequences are related to bioprofiles. Also looking at monitoring of informatics environments. So with a tool like D360, it can be connected and is often connected to many different data sources. And so we have to make sure that all those data sources are up and running satisfactorily, and we want to warn anybody before anything becomes a problem. And we're also working on integration with more of the specialized design tools. So that might be something like Live Design from Schrödinger. It might be something like Design Hub from Chemexon or Chemtrax from Elixir. All of those are really for tracking individual designs through the research process or bringing to bear more computational tools. And we want to integrate D360 with those to bring process together with data, to bring specialized modeling capabilities together with experimental results. And we can also feed data from D360 into those tools to give a better context throughout. So the steering committee is very important for our strategic piece, but we also do a lot of tactical work based on our users. So any user within D360 can actually request an enhancement through the application, and we constantly triage those in order to best develop the product in line with what users really want to do. And so we've seen our usability evolve quite drastically over the 10 years I've been involved here in order to really satisfy user workflows. So we come up with our own ideas and thoughts as well, but we balance those with input from users. When we are developing new capabilities, we do this in conjunction with our users and with our customers. And a quick example here is a new capability we just released in June this year called transformation analysis. And this will take an example core or an example substituent from a molecule and say, hey, if I keep the rest of the molecules the same, but change that substituent or that core, what happens to my bioprofile? Where do I see positive changes? And really, this is to help guide design. And the whole interface and capabilities around that were designed in very close collaboration with our customers. Lastly, we usually get together on an annual basis. Pandemics obviously affect this kind of situation, but the D360 user group meeting gives us an opportunity to have all of our customers come together and share real world experiences and develop best practices. So we have a somewhat different user group meeting to, I think, what most organizations have, where it's essentially a small amount of Certara input and a substantial amount, usually over 80% of customer input. And the most interesting parts of those are the discussions after the presentations, where people are getting better ideas of how to deal with their data. They can pick and choose, the good pieces of what others are doing. So having said all that, let's take a quick look at D360. So we're just going to flip over now and just take a very short demo of D360. So what you see here now is the D360 dashboard. So the key part of this are these little rectangles in the middle. The left-hand side gives us an idea of going to different key parts of the application. The little tools at the top right allow us to manage the dashboard. But these rectangles in the middle are what we call widgets. So this one at the middle, in the center column at the top, the CSRC Project GD, is a project-based data view. So as a medicinal chemist, I'll want to see my project data on a regular basis. So all I need to do here is this quick search widget has been built by a colleague and shared through a shared workspace. So every member of the team can get to this. But now, in order to get to my project data, I'm just going to click the search button. So once I've clicked that search button, then that's going to launch our data set. So now the query is executing. In this example, it's actually going against two separate databases, one which contains all the chemical information, the other which contains all the biological activity information. So here we just have start off with a simple spreadsheet view. So each row here is an individual chemical compound. We've got our chemical data in terms of the ID and a live structure. We'll take a quick look at some structure tools a little later, but we can do a lot of things with that structure, like R group analysis, match molecular series, and we'll look at the transformation analysis. Then we get to our assay data. So here's our kind of primary assay for this project that's color-coded. So we want low IC50 values, meaning potent data, and this spreadsheet is already sorted. So our most active and most potent compound is at the top. You'll also see some different values in here. So the main value in every cell, so for example, is 0.32, is actually a geometric mean of all individually measured IC50 tests. And we have business rules behind that to make sure that the science of the data is maintained and that we're aggregating this to providing a meaningful number. But also you'll see a couple of other values in this cell. So at the bottom left, it says 2 of 2. That just means this compound has been tested twice. And both of those values went into determining our geometric mean. If you look at the next one down, this compound has actually been measured 4 times and all 4 values were used. It might be that in some cases you have 4 tests. 3 tests gave you a determinate value and the other test gave you a greater than. In that case, the compound would have been measured 4 times, but we'd only use 3 of those values in determining that IC50. In the next column, we actually see a representation of the individual dose response curves that we utilized in order to come up with that value. We can drill down and look further than that, but the goal here is to give an idea of how meaningful is this number in terms of the quantity of measurements that are behind it. And also, if you look at the top right of the cell, it gives a little number. In this case, it's in red, and that red value is saying, hey, there's a large spread of values under here, and you should be a little careful. And whenever you see that kind of thing, we can always double click on these data values to drill down, have a look at the individual values, and also in this case, also the fitted dose response curves associated with these compounds. So now we can get a good view of not only the data values, but the quality of those data values. These other columns over here look at selectivity assays in this project. So these are colored the other way. We don't want to be active at these compounds. So low values tend to be colored red. And so here we can see on our most potent compound, that's great. It's really potent at 0.12 nanomolar in our primary assay. But when we look at our selectivity assays, it's actually pretty potent in some of those as well. So it may be highly potent in the assay we want, but it is not selective. If we look a little further over in our spreadsheet, then we can see things like molecular weight, log P. These are from the database, but if we want to add our own calculated properties, we can just say we want to add some. We provide a standard set of calculated properties here with the product. So let's calculate TPSA or heavy atoms, for example. But we can also plug into any calculations that our customers have available. So, some of the larger customers, there are many thousands of calculations available to end users through this. And when we do that calculation, you'll see at the end, we get a couple of extra columns where that calculated data is available. One column here, just a very basic one, is a calculation within the spreadsheet that uses other data. So this is an equation column. It just calculates a very quick selectivity ratio between EGFR and our primary assay. If I right click and say I want to edit this equation, we can see that it's very quickly one column divided by the other, and these are our CSRC columns and our EGFR columns. But the equation system here is very extensive, allows us to do logical operations, text handling, conversion into different log spaces, and also things like lipophilic efficiency, ligand efficiency calculations as well. So you can calculate a lot of extra pieces here without the need for, again, going out to other tools. A really key capability in this is this MPO score. I'm just going to move this up to the beginning. So we saw here on our first compound that we're highly potent in our primary assay, but we've got a bit of a problem with some of our other assays. So it might be the most potent, but it doesn't necessarily make it the best compound. That's what our MPO score is for, to say, score this compound across all of the data that I'm interested in. If we just have a quick look at this score, so we'll just edit it, it says, well, our CSRC value, our primary assay, we want low values to be good. But for all our selectivity assays, we want high values to be good. We can drill down in a little more detail here. So this is our primary assay. We're saying anything less than 10 nanomolar is good, anything above 1,000 is bad, and that gives us a score. So when we put all this together, now we're going to get a score which gives us a better profile. So if I now sort this in a descending manner, we now have our best scoring compound right at the top. And we can see that this is less potent than our most potent by at least an order of magnitude. But we can see now that we get green right across the board. So this compound is actually the one with the nicest profile. So here we've just started to look at some of the capabilities, but we can go a little further here. So maybe what we want to do is have a look at our distribution of our MPO score. So I'll just click that one and say, click our histogram icon, and we'll develop a quick histogram of this kind of data. We might also want to look at a distribution of our primary activity. So we'll click that again, generate a second histogram. We can just arrange those nicely. That second histogram of our activity data is probably best put on a log scale. So I can quickly do that and just say, pop that on a log scale. And then also, I probably want to remind myself which end of this MPO score is good. So I'll just apply the same color scheme here and say, color by the MPO score. So we can remind ourselves that it's this end of things that's good. And if you select in any one of these viewers, you'll notice it selects it all. So there are lots of visualization capabilities. One I'll show you, which is most intriguing. So this MPO score number gives us an idea of how good or bad the compound might be, but it doesn't really tell us why. So if we go, we can actually calculate what we call a bullseye plot. So I'm going to calculate this bullseye plot based on MPO score. So once I've done that, we now get a new bullseye plot. So this is what we have down here. Again, I'll just color that by our MPO score. Now, this bullseye plot basically says our ideal criteria are in the center of this. And currently no compound is in the center, so no compound is reaching our ideal criteria for this project. But it does say that this compound here, which is the closest to the center, most closely matches what we want. If we just shrink this down a little so we can see a little more data and have a look at our assay data, especially our selectivity assays. The nice thing about this bullseye plot is, well, we can see, for example, that there are some points down here which are all about, 70% equivalent to what we're looking at. So when we look at those compounds, we actually find that they're mostly green in terms of our selectivity assays. Actually, we can see this a little more clearly if I just make this a details viewer. So we're kind of green here, but we are not so good on our primary activity. So this group of compounds is 70% similar to our targets, but has a problem over here in our primary assay. If we look at a different group of compounds at the top, these are also about 70% similar to our criteria, but they have quite a different profile. So now we have much better primary activity, but when we look at our other assays, we find we have a kind of problem in our c-Abl and HCK assays. So this kind of plot gives us an idea of what kind of different profiles there are within our data set and how those relate to chemotypes. So we'll just pop our spreadsheet viewer back. Something else we might be very interested in here is, are there any correlations between these assays? So we could certainly generate scatter plots of all these assays, but very quickly we can understand, are there correlations here by looking at a correlation matrix. So here I'm just going to say, look for correlation matrix between all of our assay data, and we'll compute that matrix. So here what we see is a correlation between all our assay columns and all our assay columns. So obviously on the diagonal, you're looking at a correlation of an assay with itself, which should be 1. The deeper and darker the red here, the more highly correlated these things are. And if things were anti-correlated, you'd see a similar shading in blue. Now, because this is IC50 data, we really want to look at a correlation of the log. And here we can clearly see that when we look at our primary data, we've got a strong correlation with column 4 and column 5. And these are our c-Abl and HCK assays. So this is a primary concern in this project. So we can select those and say, I want to create scatter plots of those things. And now when we create scatter plots on a log-log scale, we can start seeing that, yeah, there is a high correlation. What we want is low CSRC values and high c-Abl values. So we want to be in this quadrant up here. So these are, kind of our best compounds. And when we look at the MPO scores for these things, we see they tend to be quite high. We can also apply filters to these things. So if we did want to filter and say we want to look at where we've got, highly active in our primaries on a log scale, we can start filtering down to that. So just want to show you a few of the bits and pieces. But if we did want to take, say, let's say take this compound, which we've already found to be pretty good, it's got a high score. But we want to understand, actually, let's just pick the best one. And we'll just filter that. So here's our best score, best compound in terms of our MPO. But maybe what I want to do is see what happens when I change this propyl group at the bottom. And for that, we can do a quick transformation analysis. So there are a number of different structure tools to help analyze and understand what's going on with SAR. Here, I'm just going to say, do a quick transformation analysis. I'm just going to sketch that isopropyl group here. We hook up to a variety of sketches. In this case, this is just going to use Biovia Draw. And we'll just sketch our isopropyl group. We'll tell it where the attachment point is. R1. Transfer that back and say, go find these series. Oops. So that's just found, hey, there are a whole load of series where there's a constant part of the molecule, but one molecule with that constant part contains the isopropyl, and there are other molecules that have something different. But this isn't really a very good view. What we'll do is complete the analysis, and what we'll get is now a view that says, when I start off with our isopropyl down here and change it to one of these other substituents, this is what I see. And this is right now done in terms of MPO score. We can say, no, actually, I want to see that in terms of my primary activity on a log scale. And so now we can get a perspective on how this alters across a different series of substituents. And also with each of these lines represents a different core structure as well. So any mouse over can show what we're doing here. The smaller plot in the top right just says, all right, I want to examine this change very, very in detail. So this is isopropyl to cyclopentyl. So that's what you see here. And we can also say, well, what happens to our different activities? So here we've said, well, actually our MPO score changes, but it's mostly neutral. But if we want to look in more detail at this, we can say, all right, well, I also want to see what happens to my primary activity. So we'll say primary activity without a tile. And here we see now when we move from isopropyl to our cyclopentyl, only 3 of these changes, example changes, are favorable. Most of them are neutral and some are less good. But we can add all of our activities. So HCK is another example. We'll add that. And again, we tend to see, yeah, it's not so good when I make that change. If, however, I select another item down here, so we'll change to this other 5-membered group. Now, with our IC50, we see, well, we tend to see less good changes in our IC50 here. But we can also look immediately at the profile over all of these things. So that's just an example of some of the structure-based analysis capabilities. But what we've done here is we've kind of built a more interesting data view that we're going to want to see with our project. So we've got a couple of plots looking at our liabilities here. We've got our bullseye map of our MPO score. I'm not so interested here in our activity distribution. So what we can do is just rearrange our viewers, get them how we like. So correlations at the top, other data at the bottom, and with our spreadsheet at the top. Now this is a much richer, better data view from a decision-making process. So what I can do is create this data view and share it with my colleagues. So I'm just going to go to File, say Save. I'm going to save it to a shared workspace, so it's available to colleagues. I'm going to save it as a new query with an imaginative name. And then when I've done that, that's now going to create me a new dashboard widget. So I close this data set. Now, maybe I come into, and we'll close that one too. And then when I come into work tomorrow, we'll see, okay, look, I've got a new widget, and every team member has now this widget on the dashboard. So if you want the basic data view that we started with, you can just run the original widget. If you want the richer view, we can just click the search. And all of that work that we did in the arrangement of viewers is now done for us. And so we start off on a much richer perspective in order to understand what's going on here. So here, everything is done for us. All our data is well organized. All the plots are there, including our transformation analysis on the separate tab. So that's really where we end up. Most usage of D360 is utilizing a widget with that one-click operation. I think it is important to show how that was built to begin with. So we started off with just a basic spreadsheet view. But somebody needs to say, well, what's in that? So we can start off by building that by saying, I want to build a new query. And so all users of the system can build new queries. In reality, we find about 25 to 30% of people do this. Another 25 to 30% of people are happy to edit these, and everybody else really just wants to consume. But it can be done by any scientist. So here what we choose to begin with is just, well, where do we want to start from? So if you're in the small molecule domain, you might want to build a compound level search or a batch level search. Or if you're a biologist, you might want to do an assay level search where you're looking for different assays. If, however, looking at biologics, don't pay too much attention to the exact words here, but you might want to look at different antibodies or different cell lines. If you're in peptide discovery, that'd be another tab here. And this interface here just varies from organization to organization based on nomenclature and the kind of things they want to look at. So here we'll build a small molecule or a compound level search. When we do that, we get a search interface. It says at the top, this is a small molecule search. Below that is what we call the data catalog, the supermarket of data. So this is all the information we can add that comes from one or multiple sources. So if we want to know our compound ID, we'll drag that over into our query. If we want to know the structure, we can do that. There's a whole load of calculated properties available in this database. Great, we'll add those over as well. So now we've started to build our query, but the important stuff is the assays. So I want to see my assays. Those are in a folder called in vitro results in this example, but this catalog is always customized per customer based on how they want to see it and how they can find stuff. So if we want to look for that CSRC assay, that'll be in enzymes, it'll be in SRC if we look down here. So here's SRC, and we can find out our assays. On the other hand, I don't want to look through that tree, so I can just filter and just say find stuff with SRC in it. Here is our folder again with SRC, and here's the assay we're looking for. So we can either browse this or search just using text, or you could actually provide compound IDs, and it would show you all the assays that those were tested in. If we want a little more assays here, we can just search for the word recombinant. And now we see everything that we're interested in. So here's an EGFR assay, here's our SRC, here's our HCK. Once we've done that, we'll just bring those over into our query. It says, hey, these three assays have the following results. We'll say we want to see our IC50s. And so now we've added in our information. So I'm just going to say here, well, it's not just the IC50, we can add other results as we want. But the last piece we need to do here is say constrain the query. And here I'm just going to say, well, these are the important assays for my project. So I want to just get everything that's been tested in those. But I don't want everything that's been tested in all of them. They must have been tested in any one of them. So I'm just going to go down here and change my query logic, just to say all these things together. So now essentially I've built the basics of my query. We can go and search this, and then that will come up with our spreadsheet. Now, often you find that some assays have terribly long names, and we can go and do things like, rename this assay, or we can rename multiple fields at the same time. So we'll just call it HCK. We can move these columns around as we want them. We can apply color schemes very quickly. We can apply a different color scheme to this, and we can build out whatever we want to see, and we can get a sort order that's appropriate. So we've essentially got our basic data view. Now that's created, we can again go and share that with our colleagues by saying save it as a query, and we'll just call it new view and save that back. So now we can close our data set, and every member of that team now will have, I save that too. Essentially every member of that team now will have that new view. And sorry, I apologize, it should have, forgot to check a button. So there's a new view that's available to all members, and they can execute that. So up till now, I've just focused on small molecules. Just very briefly, we can deal with other kinds of modalities. So here's an example of some peptides with sequences. And if we so desire, we can do a sequence alignment of these peptides. We can color and format these. So we can start getting an idea about how they align. Just another example here is preclinical safety. So here we're just interested in how the body weights of animals appear over time. And these are just examples of different kinds of data that we can bring into the system. So I'll just conclude the demo there. There's a lot more to talk about if time allowed, but if you're interested, please, I'm very welcome to get in contact. So let's just switch back very quickly to PowerPoint. So just in summary, really what we're trying to achieve here is make data highly accessible. And we want to make data highly accessible so we can speed up these design, make, test, analyze cycles. But because we've got consistent treatment of the scientific data, we also want to make it so less cycles are required. Very important. We are definitely built for scientists, right? We have a distinct focus on helping the scientists perform their workflows to add that value. We want to make research far more data-driven than it is if you have difficulty accessing your data. And we work with our customers very closely, both strategically and tactically, in understanding what they need and determining product direction. Very importantly, both in terms of how scientists can access their data, but also, just in terms of the logistics of deploying such a system, is we want to keep the maintenance cost very low. So when we deploy D360, we configure it to access the data. We build that data catalog so scientists can be self-service. But beyond that, D360 will automatically make available any new data. It will find new assays and make them available in the tree. So at our largest customer, which is multiple thousands of users, essentially the maintenance of the system is a fraction of an FTE. And the last point here is, we actually started off with larger customers here. So, we have great experience in deploying D360 where it's accessible across multiple sites globally. But those same capabilities that allow us to do that and have that available at a very low maintenance cost also are very attractive to small organizations where you might think that this kind of system is difficult and complex and only for big pharma. Well, not anymore. That's a thing of the past. So just wanted to conclude there, but happy to answer any questions that people might have.
Great. Excellent presentation. Appreciate that. Let me do a couple things here real quick. Number one, I want to go ahead and get the recording stopped.